Well, welcome everybody to our Glass Brain interview today. And we have with us Dr. Peter Fox, our Glass Brain Award winner, who accepted his award this morning. Before I start posing questions of Dr. Fox, let me just give you a brief bio in the unlikely event that you might have missed this morning's presentation of the Glass Brain Award. Very briefly, Dr. Fox has started his education out with a BA in Liberal Arts from St. John's College in Annapolis, Maryland, followed it up with an MD at Georgetown University School of Medicine in Washington, D.C., and then went on to do two medical residencies. The first one was in internal medicine, and that was at Duke University Medical Center. And the second one was in neurology at Washington University Medical Center in of course, St. Louis, Missouri. He is a academic clinician and a researcher who has had immensely interesting training. He started his formal research out as training on an NIH T32 award. And that was through the Washington University Department of Neurology and Neurosurgery. And the idea there was to develop novel methods and apply them to mapping sensory motor brain function. And these methods, of course, were oxygen-15 positron emission tomography, PET, as we know it today. He then went on to a KO7, an NIH KO7 Clinician Scientists Training Award, and the idea there was to now move these PET methods to higher order brain functions, such as language and directed attention. He has held multiple academic positions, first off at the Johns Hopkins University School of Medicine, where he was a founding member of the Mind-Brain Institute. He has been at the University of Texas Health Science Center at San Antonio since 1991 as the founding director of the Research Imaging Institute, and he's still there today. And this unit is dedicated to the development and application of advanced imaging methods. He is also the Vice Chair of Research and Research Education in the Department of Radiology there. And he holds the Malcolm Jones Chair of Radiology. And he's a professor in the Departments of Neurology, Psychiatry, and Physiology at UT Health San Antonio. He is all over the map on the graduate faculty side. He participates in engineering programs, scientific programs, academic programs, and also medical programs and radiological ones in particular. And overall, he's trained 31 pre-doctoral trainees, 13 postdoctoral fellows, 17 faculty under these US federally funded faculty training awards. And he is the tour de force behind the journal Human Brain Mapping. He founded it in 1992 and now has 3 deputy editors-in-chief to assist him, and they are Jack Lancaster, Gary Egan, and Simon Eckhoff. So welcome to this session, Dr. Fox. It's lovely to see you.
Thanks. It's very nice to be here. I appreciate the opportunity to spend some time chatting with you about OHBM.
Well, yeah, and then let's talk a little bit about that. And in fact, you were the instigator, of course, of the human brain mapping workshops in the late 80s and early 90s in San Antonio, which brought in all of the senior neuroimaging scientists and neuroscientists at the field at that time, and we're all getting together to create a common 3D standardized imaging space, which was a novel concept at the time, and also wanted to share neuroimaging data across labs. And so we had like a proto-open science initiative back then, if you allow me to say that. And these workshops sowed the seed of the human brain mappings, human brain mapping meetings held in Paris and Boston. They were the first ones, which of course you also helped organise. So how did you imagine the future of OHBM at the time?
When we were first starting this, actually, I organized a meeting when I was at Johns Hopkins shortly after I arrived there, and wanted to pull people in from different disciplines. The unimodal, like the neurology meetings, the psychiatry meetings, just the meetings within your training discipline seemed to me really inadequate because we all have to work with computer scientists, with mathematicians, with statisticians, and they don't come to the discipline specific meetings. And so I wanted to start making meetings where people from different backgrounds who were interested in brain mapping could come together and meet. And so the first one was at Johns Hopkins, and that was really fun. It was about 100 people, and Albert Guetta came to that and spoke. Jack Beliveau came to that. David Kennedy came to that. There were people that were both established in the field, but like turning toward neuroscience, and other people that were just getting started. And that was really fun and successful. And so when I moved to San Antonio, I knew I wanted to keep that momentum going. And so I started putting in NIH grants to fund these targeted meetings, and the NIH responded very well to that. So they kept giving me year after year funding to do it again. And so I'd say, okay, that one worked, and here's who came. And after a while, we bounced around a little bit on the format, but we got pretty well organized, and we always had a session on databasing and data sharing. And we always had a session on spatial normalization. And so I was working with 3D, with volumetric data, but people that were working in 2D space came right away. So David Van Essen came, and Anders Dale came, that people were showing alternative spaces. And so we all talked about that. And so we brought in people from other disciplines that had done large scale sharing already, from genetics. And there was a really interesting project from the Child's database, where they were then and still are sharing natural language acquisitions across developmental ages. And so there were these prototype data sharing initiatives that were out there. And so we were all discussing how we could structure what we did to promote data sharing at that point. And so there was really pretty strong consensus that this was a good idea. And that kind of naturally led to making it bigger, because I was funding 200 people. And I mean, we had a specific structure that spread the money really far, but that was real successful. And so we began talking about how to make it bigger still, and make it international, and get it, out of San Antonio, or not exclusively in San Antonio. So it just kind of emerged from that structure and that core idea that we needed many disciplines all meeting together to make this kind of thing work.
Yeah, and of course, you had lots of scientists, from Europe as well in that venture, and so it was always a multinational thing that seeded that cooperation. So in some ways, it's not surprising that there was so much buy-in, because it was inclusive of people from various disciplines and various countries as well, right?
Always from the start. Really, I was trying to bring in the people that were leading in each area. And so I'd been doing early versions of statistical parametric imaging, but then the Hammersmith group started working also in 3D space and doing statistical parametric imaging and releasing software. And so they, of course, need to come. And so Friston for Koviak came, Bernard Mazier and Per Rowland came very early. So there were really, that was my goal, was to bring in the people that were at the cutting edge and have them all talk to each other about how we needed to move this forward, and what would be the best approaches.
Mm-hmm. Well, we'll come back to that in a little while, but it's fascinating to hear how that all started, of course. But I'm going to take you way back into the past, about when you grew up, and where did you grow up, and did you have siblings?
My dad was a physician in the Navy, and so we grew up wherever there was a Navy base with a hospital that my dad was assigned to. So San Diego, Cambridge, Annapolis, Philadelphia. Eventually, I spent a lot of time in Washington, D.C., because he was assigned to the National Naval Medical Center in Bethesda, so right across from the NIH. I spent 6 years in Boston, so that's, yeah, kind of peripatetic, the way the military is. And yes, a lot of siblings. There were 6 of us total. 3 of us are physicians, and so it was a pretty tight group. We all still Zoom on a monthly basis, even though we're distributed across the map now.
Well, that's pretty good. Out of six Navy brats, 3 went to medical school. That's a pretty good set of statistics. Yeah. And so what did you want to be when you grew up?
We lived on the hospital base. Mm-hmm. And so the people living next door on either side were, like, one was a psychiatrist and one was an internist, and my dad was an interventional cardiologist, and like everybody I knew was in medicine. And so I just kind of assumed from the start that I'd want to do medicine. My mother was a pediatric intensive care nurse. Oh, wow. So yeah, I just always wanted to do medicine one way or another. It took me a while to figure out exactly what style of medicine I wanted to do, but that was a real early choice.
Yeah, that was going to be my next question. When did you figure out what style of medicine you were going to pursue?
About halfway through college, and in the sense of doing clinical neuroscience, it was just always a toss-up between neurology, neurosurgery, and psychiatry. And eventually I gravitated toward neurology, although I came close to transferring into a neurosurgical program halfway through residency. And so I got an offer, and it's like, oh, do I take this offer or not? That was a tough choice, figuring out whether neurology or neurosurgery.
Mm-hmm. Yeah, a lot of clinician scientists still struggle with that choice today, in fact. Yeah. And so when did you then, what pushed you into research then? What you crossed to the dark side? What did that for you?
I really always expected to do academic neuroscience. I only looked, when I interviewed for residency, I only looked at programs with training awards for research. That was like first cut. And that advice came from a mentor when I was in medical school. Stanley Cohen was a neurologist, and I was talking to him and doing some work in his lab and asked, how do I filter through all these programs? And he said, well, here are various ways you could think about it. I actually applied only to programs that were doing imaging and that had T32s. And so
you found a mentor early in the piece who put you on the right path.
I did, yes. Actually, he was doing stroke research, and so it's in the ballpark. I didn't think I wanted to do stroke research. I'd actually been reading some of the papers that were starting to come out of Denmark, Ingvar and Lassen, and the pre-PAT
Mhm.
activation studies.
The xenon studies.
Yeah, and thought the xenon studies and saw that that was real interesting, and wanted to know where I could do that. And so actually, as a medical student, I interviewed with Mark Rakel and told him, I want to do brain activation studies. Could I do them here? And how early could I do them here? And could I do them during residency? And when I was told yes, yes, and yes, it's like, well, that establishes I'm listing St. Louis as number one. And so
That's great. And so at the time when you did go to St. Louis, before you started the O15 studies, what were people doing xenon inhalations at the time in St. Louis as well? Or
no, not at all. That lab really has a long history. That Terpagosian was the one running the division, and he really had a tremendous vision, and he was the one that selected O15 as the target, and got the first medical cyclotron in the US. So he had a cyclotron in the basement, and he was building PET scanners there on site. And so they went through a series of PET scanners. They had one that was just a single ring, and then one that was 3 rings or 2 rings. I saw that one when I interviewed, but that one was gone, and there was a new one by the time I came to residency, and that was the PET 6. That had 6 rings. And so that was a pretty interesting lab. They had engineers and computer scientists and physicists, and in that space they had neurologists and psychiatrists. So Rakel's office wasn't in neurology, it was in the Mallinckrodt Institute of Radiology. That's where he's at. And Terpagosian talked to people all the time about scanner design and which crystals he was using and how he was how big would the aperture be. And so the environment was very interactive, that he was getting input from clinician scientists, from Rakel and others steadily. And so that to me was an obvious template for how to do good research with imaging technology. And yeah.
so he was another person that you looked up to.
Oh, yeah, very much. He knew that he wanted to do oxygen 15 so that he could do carbon monoxide, he could do oxygen, and brought in Rakel well before I got there. But Rakel had worked in Cornell and had done basically whole brain activation studies, breath holding studies, gas manipulations with Fred Plum. And then served time in the Air Force, and so knew high altitude physiology. And so he came in as the oxygen and blood flow physiology expert, and then worked to get these methods up. But it was within a framework that had already been created for producing oxygen 15.
So he certainly had the expertise. He understood the basic physiology like nobody, particularly with all of those experiences that you were describing that he'd had. It's amazing, really amazing. So have you had a PET scan?
Have I? Many.
Yeah.
Yes, quite a few. Yeah, I've got some great photos of like the first activation study that I did was on me, and it was wearing those goggles to see if we could get rate effects and to make sure that all the procedures worked. And so that was with the art line, that was fully quantitative. And then I got the data. It's like, this is me and my data, but I will figure out how to make this work and how to analyze it. So
yeah, and so it must have been quite a challenge getting all of the equipment set up for those activation tasks back then, huh?
It was. I learned things that I never expected to have to learn. The visual stimulation systems were, well, there was one from Princeton Graphics, which had a very high resolution screen that we could program. So I had to learn how to program visual stimuli on this dedicated image processing or image display system. Much more complicated than what we have to do now. But you know,
so you were programming in octal or hexadecimal?
In some things had scripting languages, and so I had to learn various scripting languages. But the resources were amazing. That, you know, if you figured out what you needed and then asked for it, generally you got it. So you know, it seems kind of fun to me now that people are talking about or are doing real-time fMRI and getting very fast processing. We had processing, so between studies, so these are 2-minute studies that are 10 minutes apart, we had the data analyzed and displayed while the subject was in the scanner, that it was reconstructed and up and subtracted. And so we knew whether we were getting effects before we let them off the table. So,
right. And the computer probably was the size of a room.
The whole lab ran on a single supercomputer with dumb terminals everywhere, except for the display. And that was a dedicated display system that was a Ramtech. And that was like a small refrigerator, and it had one monitor. But it was there and it worked.
When we think about it now, isn't it? Because I look at my cell phone, and I just replaced my cell phone, and I think about the computing power of that. And I think about the computing power in the old days of the computers and at the time. And it's absolutely mind-boggling to think about what we have now and what we can do. But even then, when you think about what people could do back then, those studies were pretty amazing. So let me ask you about that. I mean, you've seen the methods and science evolve over these decades. And what do you think, looking back over all those years, what did you find was the most challenging things to do that you experienced? And why was this the case?
One of the early challenges, when I was trying out different task types and looking at what we could explore and not explore, and trying to figure out how quickly we could push the envelope up toward cognition. Motor tasks were straightforward. You could get PET results in a single subject. Visual tasks were just very easy. Somatosensory worked immediately. We had some good hints because there were very good people on site. So when I wanted to know what was the best somatosensory stimulation, I got to go to Harold Burton, who was doing somatosensory stimulation and recording in cats. And he said, well, you need a piezoelectric vibrator, and you need to go at these frequencies, and these are the receptors that you're going to get, and these are the regions you should expect to see. You'll see S1 and you'll see S2. And it's like, thank you very much. Here's a roadmap. And so that's what we did. And so that kind of advice of going directly to animal physiologists and then importing those stimuli was extremely useful. Mm-hmm. But what was really difficult was getting directed attention effects. Language was very straightforward. That if you had put people in the scanner and had them do language tasks or present word stimuli, you right away saw reasonable effects. But attention tasks were really tricky, and it took a good while to figure out how to get attention modulation that really made some sense. And that one was slow.
So that would have been the most challenging thing you would have experienced in having to do, you'd say?
That had the most strikeouts. We took a lot of swings without connecting before we finally got a robust attention-related activation effect.
And that brings up a very important point. If at first you don't succeed, you try and do it again, and that's a very common story in science when people are developing things from scratch. So it's good to hear someone who's had so much success talk about, you know, some of the challenges that you have experienced. And so you've been in the lab for many, many years. Do you have any favorite anecdotes? Because funny things happen in the lab. Do you have anything that you want to share or that you can share with us? That still tickles your funny bone years later.
So I don't know if this is so much funny as informative and memorable. We were doing preoperative mapping tasks for the Department of Neurosurgery, and had been working early on with Mike Posner and starting to do some of the language papers that eventually have become very well known. And one of the tasks was the verb generation task. And we had a patient in the scanner, and we were doing this immediate subtraction that I was doing the intervention and then checking, and there was no effect. He was doing the task, and I'm looking, and it's like, nothing's happening. So I asked then a postdoc, but now a professor, Jose Pardo, who was working with me. And I said, Jose, did you do anything different? And he said, no. And he was doing really good before the scan. So what do you mean? He said, well, I practiced him. And I said, okay. And did you change the word list? And he said, what do you mean? I said, well, we have different word lists. If you show him the same word list, he won't ask anymore because he'll know the words. And so he'll just do it by rote. And he said, oh, no, I didn't change the word list, right? So it was pretty impressive that the effect immediately went away as soon as they'd seen it before. So he wasn't doing a somatic judgment anymore. And so that turned into a paper that now is the practice effects paper that's also very well cited. And it was like, that was just a big mistake. But it's like, but it's a replicable mistake. We can make this come and go. And so
that's brilliant.
We weren't looking for that at all. But when we found it, we knew we found something. And so
that was an important control, because the control tasks, of course, we all argue about those tasks. The active tasks are easy, right?
I had just kind of assumed that we shouldn't practice people on the word list. So I'd made up multiple word lists. But if you ask me why at the time, it's like, well, it doesn't seem like a good idea to show them the same word list over and over. But Posner later explained it. And he said, well, you've gone from a somatic judgment task to a paired associate recall task. Those are completely different systems. It's like, there you go.
So you live and learn with happy accidents. And sometimes good things like that happen in the lab. Okay, so thinking about you've got so many highlights in your career. What one would be the most precious to you and meaningful to you, and why?
I guess the biggest sigh of relief came with the bold effect, replicating and confirming the uncoupling, the task-induced uncoupling, right? That you get blood flow and glucose metabolism going up and oxygen metabolism not going up. And I've been told so many times by so many people who were very well respected in the field that that was obviously a mistake. and that I should go back and check my data for the 100th time and make sure that, find where I'd made the error was generally the way it was put. It's obviously an error. And so you just haven't tracked down the error. It's like, boy, I've looked at that every way I could look at it. And so when the bold effect came out and the papers describing it said specifically, we were doing this. Excuse me. Sorry.
That's okay. It's so you were saying about the bold effect coming out.
That the papers that came out specifically said we looked for this effect because of the PET papers, and we found it, and so this confirms those observations. And it's like, all right.
Yeah, I can imagine it would have been a sigh of relief, because you always, when you put things out in print, even though you replicate them, you always wonder, well, is this completely true, even though you've checked everything many, many times? And so that was, I could understand your sentiment about you feeling vindicated, you felt relieved. And I can say, I think the bold field in general gave you a big thank you, because that really helped the bold field along. So that was mutually beneficial, in many ways. So let me think. If so, you've had all of these really interesting research experiences and positions. If you had your time over again, what would you do differently in your career? Anything?
Yeah, I don't know. I was going to say maybe keeping more time in clinic, because I haven't done clinical work other than clinical trials. So I see patients through clinical trials, but I don't have my own regular clinic, and haven't had that for a good while. And it's like, yeah, but that was kind of fun. that was pretty interesting, seeing patients and hearing their problems and trying to help solve them. I guess I kind of envy, I work a lot with MD, PhD students, and they're getting into clinical medicine and seeing that they're having that fun, but I'm not. And part of that's because when I started doing this, neurology departments didn't quite understand why a neurologist would do imaging research. And there just weren't that many people doing it. There were some, but not many. And so the neurology departments, both at Hopkins and in San Antonio, were like, well, why don't you work in radiology? It's like, really? I'm a neurologist. And they're like, yeah, but you're a different kind of radiologist. So, I mean, a different kind of neurologist. So, yeah, I think I should have pushed harder on that. So,
well, yeah, I can understand why I used to test patients and map invasively as well. And so you learn. For me, the things that I learned the most about the brain came from that side of things. So I understand exactly what you're talking about. But when you think about it, it's interesting to see, you mentioned MD PhDs. There are some amazing MD PhDs at the moment in the world. And I think the one that makes me think the most is Carl Deseros, who still sees patients and is the, doing all this optogenetic stuff in animals. That's astounding to me.
That are one of the faculty at the Health Science Center. Francisco Figueroa is a pediatric transplant surgeon who became dean and then became president and then became chancellor and then came back to being a transplant surgeon. When I talked to him, he said, no, he always made time to keep up practice, even if it was on weekends, or he would take slots from other people around his day job. And so he figured out a way to do that. And
Yeah.
so,
Yeah. So that was his passion. He was practicing his
yeah.
passion. And it sounds to me like you've done that too, even though you said you perhaps might have taken more of a clinical track. It sounds like you've been practicing what you're passionate about as well.
That's certainly true, that using these tools to really understand as much as possible about the human condition, about how our minds work, how we're organized, how does the biology make the psychology happen? That's really pretty interesting. And I am working with students, trainees who are really focused on trying to move this or continue to move it into clinical practice and to figure out how to make diagnostic and therapeutic biomarkers. And that's pretty interesting, trying to change. I guess one way I look at it is, instead of trying to change or improve health one patient at a time, it's like, let's try and do it one disease at a time. Let's find a biomarker for this disorder and try and change clinical practice in this condition.
So that's, I was just going to ask you with what current directions your lab is taking that you were really, really interested in, and you went there. So is that what you're actively working on now?
Yeah, very much so. And trying to hammer out a pipeline for discovering biomarkers. And really what we're doing is continuing to work with the meta-analytic algorithms that I've been working with, since the 80s, and use those to find disease-specific effects. So where are abnormalities associated with post-traumatic stress disorder or with Parkinson's disease? Where are these structural and functional abnormalities, and do they have connectomic abnormalities? And which combination of these effects is the most sensitive biomarker? Does it correlate with clinical symptoms? And so building them meta-analytically, so for the whole population, and then moving them to the individual level. And so I think that actually can be basically a pipeline for biomarker discovery.
Mm-hmm. So have you been affected tremendously by the COVID pandemic, or have you got so much data that you're sitting on that you can mine it? How has that affected your lab?
It slowed down data acquisition some, but not tremendously. And that we set up protocols pretty early for testing people, looking for symptoms, continuing to screen them and move them through and scan them, wearing masks, but testing them for COVID. And so it didn't slow things down as much as you might expect. We were given in our school the ability to continue running either the clinics or the labs, and the policies. We were given general guidelines and a framework, but within that, it was left up to the individual institute directors and department chairs. And so it was pretty effective, I thought. I thought it was very well managed.
Mm-hmm. And of course, the American College of Radiology had their guidelines as well, which everyone was looking at. And it's interesting, though, there are some interesting papers coming out about long COVID and potentially effects on the brain. And that might be something that people here in neuroimaging might be looking at in the future. Have you thought much about that at all? Or
very much so. We've got actually a U19 in review now, specifically looking at that. And the imaging studies that have already come out are not very large numbers of subjects, but they give pretty strong evidence that the chronic effects. I mean, the acute effects of COVID look like hypoperfusion and, you know, ventilator damage to the brain. And so strokes, hemorrhages, small vessel occlusions, things related to the acute immune-mediated responses, cytokine storm. But the chronic effects are definitely being documented. And there is an increased risk of dementia late. And so there have been some large studies that have come out that have shown that if you have COVID, your likelihood of having late subsequent onset of dementia is substantially higher than other respiratory disorders or other infectious disorders. There's a paper actually that just appeared on bioRxiv that's from the UK Biobank data. And that's showing very much the same thing. And that all looks neurotropic, which is what we've been expecting and what the early papers, mostly PET papers, showed, that FDG PET is giving an olfactory plus, like an extended olfactory network pattern with olfactory bulb and piriform cortex and amygdala and hippocampus, and then to the limbic system. So I think, I mean, obviously these are regions that are going to produce dementia and emotion dysregulation. So I think this is an area that needs a lot of exploration, and it's starting. We're hoping to acquire more data on that, but there are a number of projects that are going on. I know the Framingham study is calling people back that have got COVID and comparing them to their previous scans. So there are things like this that started up late fall last year, maybe late summer.
Yeah, this is important. This is going to be an important growth area in neuroimaging, an unfortunate growth area for increased impairments in cognitive function. But I think there'll be a lot of work looking at cortical circuitry and networks that will come out of that, I would imagine. So that's great that your center is in the thick of that. I'm going to switch gears now, because I want to know how, after a hard day's work in the lab, you unwind.
So probably the most, the daily thing is I run and swim. And so I actually live in a really lovely area. I live right on a golf course in the middle of the medical center. They built our medical center around a golf course. Don't ask me how that came to be, but so I get to go out and run on the golf course in the mornings, and usually get a chance to swim several afternoons a week. So that to me is the great way to unwind. So,
is it meditation time for you, or what?
Yeah,
are you focused?
I never play any music. I just listen to the birds. There are deer out there. There are herons. There's hawks, and so I really just like to look at the nature while I'm out there.
So what advice do you have for maintaining a work-life balance then?
Play a lot.
Play golf a lot.
Or just in general, play a lot. I like to ski, and so and hike, and so I never miss those opportunities. I always Shanghai my wife and daughter to go to. I mean, we went skiing in Taos in the middle of the pandemic and just drove up to Taos so we didn't have to fly. And we went to Alaska and went hiking. I actually just got back. I went up to Seattle, and with a former trainee, we rented enormous motorcycles and drove up to Mount Rainier and went hiking in the snow on the glaciers.
Oh, wow.
There's still snow up there. They just opened the park. And so that was real fun to go up there and go hiking in the snow. So like that, just, get out
That's
of
great.
town, do fun things.
That's very good advice for people who, particularly after this last year where we've all been stuck inside with COVID and we're starting to emerge. So this is good to hear. Now, I'm going to ask you another personal question because for all of the decades I've known you, you're such a dapper dresser and you have probably about a zillion bow ties. How many do you have, and do you have a favorite?
It's less than a zillion, and it's probably more than 150. So it's somewhere in that range. That actually, I do have a favorite, but I also have favorite shops. And so I always would look for places. It's not that easy to find interesting bow ties. And so I would look for places I could go. So I used to try and route my flights through Atlanta because the Atlanta airport had a shop with really good bow ties. And so every chance I would get, I would fly through Atlanta and make sure I had a long enough holdover to spend at least an hour looking for bow ties. Right. And some of the nicest ones I've got, which I don't know, I've had them for 20 years, I got in Piccadilly in London because I'd heard that they had very nice men's clothes. And so they did. And so you could get very, very lovely handmade bow ties in Piccadilly. But beyond that, my favorite is one I got from my dad, who wore it when he was an intern at Hopkins.
Oh.
And so when he was in the Navy, they wouldn't let him wear bow ties. But you probably know this, that on the East Coast in particular, neurologists wear bow ties, right? And so when it's like, well, if I'm going to be a neurologist, then I ought to start wearing bow ties. And so my dad gave me that bow tie. He said, this is one of mine. I wore it when I was an intern, but now I have to wear the straight black Navy tie. So
interesting. Yeah, that's an interesting story. Well, in Australia, Australian neurologists wore grey suits. I don't know whether neurologists had a particular colour for suits, but it was the grey suit. Just so that in the Southern Hemisphere.
All right. Then I'll be able to pick them out in the crowd. It's funny that at OHBM, I always make a point of wearing bow tie, right? And particularly if I'm doing anything formal, if I'm on committees, right? And so people can always find me. That there's just never any doubt. It's like, you're Peter, right? It's like, yeah, right. If I take off the bow tie, people are much less certain. And so it's an easy way to go incognito is just to not wear a dress shirt and not wear a bow tie. And then people
when you were in Honolulu that time.
In Honolulu, but it works at any OHBM meeting. If people are not sure who I am if I don't have on a bow tie.
Okay. So just remember, all of the I have no idea how many people are listening to this, but they've made a note of this. So you're a marked man now. All right, let's turn to the future in the short time we've got left. Talking about scientists now, young scientists coming up, what sort of challenges do you see for them that you didn't have when you were building your scientific career?
It's a much more crowded playing field. In the 80s, when we were starting to do this work with O15 PET, there really weren't labs yet that could compete with us. And so almost everything we could do, we could publish in a high impact journal. And so there was a remarkable chain of papers, first of this, first of this, first of this. And so that opportunity is not so available. But one thing that I see that makes me jealous is that the techniques that I think we can all see pretty quickly should inform clinical practice. So we're doing language mapping. Well, that should be preoperative mapping. And right away, the neurosurgeons in St. Louis were right on that they understood that and they wanted that. But it took 25 years for it to become part of clinical practice, right? It took a long time. But now the disciplines are getting well informed enough so that I think this kind of computational imaging, computational neuroscience using imaging, that the people being trained right now and going through residency and into fellowship, they can change things, I think, much faster. And it's going to be up to them to change, particularly radiology and neuroradiology, from a discipline where you look at images to a discipline where you process images and compute parameters from the images, the way that people have been doing in, say, renal physiology and cardiac. Cardiac MR is highly quantitative. They want to know stroke volume, they want to know wall motion, they want to know numbers, right? And so now we can give numbers to brain function and can compute network coherence. And so I think the neuroradiologist and associated neurosurgeons, that they can bring this computational power into the clinic now and can change medical practice, which is very slow to change, right? But I think we're in a position where the work, I've been doing it as basic science, hoping eventually to see it impact medicine. And they can do it as medical science and see it change how people practice. And so I think that's really a very valuable change in paradigm. And I'm really hoping to see that realized soon, like this generation should change the way neuroscience, clinical neuroscience is practiced.
Right. And you mentioned earlier when we were talking about these big data sets and UK Biobank and you know, the Human Connectome Project, these are all very large undertakings that have never been seen before historically. And I think people now can mine the data in ways to pull out interesting things that, as you mentioned, that preprint that came out the other day, I had a look at that as well on long COVID. That was out of the cohort from the UK Biobank study. So these are opportunities that never existed before, but they take a different set of skills, as you say. Computational neuroscience is very, very important at this point in time. So I was going to ask you about what you feel the big game changer is going to be over the next 10 years, but I think you've already gone there in a sense, right?
Yeah, I think the challenge is going to be having clinically trained neuroscientists or teams working around a clinical neuroscientist, because it's never one person, right? It's always groups of people, but with the goal explicitly in mind of moving this into clinical practice and understanding how to get FDA approval and how to get CE approval and how to meet the regulatory challenges and how to interact with companies, like whatever it takes to get this into the clinic, not just a published paper saying we saw this interesting effect, but to want to change medical practice and to have that be an explicit goal of their careers.
Right. Yeah, and just to we've got about another 5 minutes or so before we finish up. But so you were saying that brain mapping is done in teams. So you wouldn't say that there was such a thing as an ideal brain mapper in the 21st century?
I have a bias there. I guess I'll apologize for it a little bit, but not too much. I enjoy training physician scientists a lot. And I think the MD background gives you such scope that you need to know pharmacology and physiology, and for the clinical neuroscientists that you know neuroscience and neuropharmacology, you know a lot of different things that are really hard for physicists and engineers to really get exposed to that in their training. And so I think the team hinges around a clinical neuroscientist, and their job is to get familiar and comfortable with engineering as a discipline and physics as a discipline and the basic scientists, that they need to reach out all around them. But I think that for this really to have the impact we want, it needs a clinician at the middle, and then but somebody who is adventurous enough and confident enough to reach out to all these other adjacent disciplines and pull in what they know. And that model I saw working really well with Terpagosian and Rakel, because they were always reaching out to basic scientists. And to when I had a question about statistical methods, they said, go talk to Jerry Cox. He's the department chair of statistics, and he'll help you answer that question. And they were just always go to the other disciplines and get them on board. Don't try and solve all of this yourself.
Right. That's the thing, knowing who to ask, having that background knowledge and having that big picture view and knowing who to ask, right? All right, so I have a couple more things to ask you as we finish up. And you are, of course, part of the OHBM, the 25th anniversary symposium, which the late Carl Zillis fondly called the Dinosaur Symposium. And I sat next to you at that, I remember. And so what were your feelings and thoughts that were going through your head at that time as you sat on the stage?
I thought how much fun it was that a vision shared by a reasonably small group of people back when we were starting up has proven captivating, engaging to so many people. And to see the scope of it, the breadth, the continued interest, and where this can all go. And so that's really quite fun. And it is based on team science and willingness to collaborate and willingness to share data. And it's exciting. It's a concept and a way of doing science that really has a lot of impact and a lot of longevity.
Mm-hmm. And then as a final parting shot, what are your thoughts, and looping right back to what we started with, what are your recommendations for the future growth and development of OHBM?
No more virtual meetings.
No argument there.
Yeah, I think all the recording of all the sessions and sharing of all the sessions has been going on for a while, and that's really quite a valuable resource. But the in-person interactions are really important. So I don't know if we need to change things. There are a lot of problems to solve, but I think I'd lean more toward pushing as much as possible into clinical disorders and trying to solve problems interacting with neurosurgeons and tackling those difficult intraoperative recording data sets. I mean, there are a lot of challenges that are just a few steps ahead of us.
Right. And that's it's a nice way to finish up this interview, because 40% of our abstracts are still clinical abstracts at OHBM after all these years. And that is, I see that continuing. And that's a really, really wonderful thing that we continue to see. So I would like to thank you, Dr. Fox, for your time today and for sharing not just your professional stories, but sharing some of your own persona as well. It's been an absolute pleasure. Thank you.
Thank you very much. Nice talking with you, Aina.